So, good evening, dear participants. Greetings from decodelife.org. So, my name is Parveen, coordinator for this workshop. First of all, I would like to thank you all for participating in this workshop. Second, myself on behalf of decodelife.org would like to express our appreciation to Ensemble outreach team for generously accepting our request for a joint workshop. Third, before starting this workshop, I would like to tell little bit about Decode Life. Decode Life is a MSME status institution registered with both Government of India and Government of Haryana. The institute has a mission to provide affordable bioinformatics education, and in this direction, so far we have trained around 3,000 people across the globe. So this workshop is another avenue through which we move a step further to liberalize bioinformatics education. We hope you all will enjoy learning and looking forward to receive your feedback. Now, I would like to invite today's instructor, Dr. Michael, who is currently working as an Ensemble outreach officer at EMBL, United Kingdom. So, without wasting too much of time, I am handing over session to Dr. Michael. Over to you, sir.
Thank you very much for this lovely introduction. Let me just share my screen. So I hope everyone can see my screen now. Welcome to the Ensembl Browser Workshop. Everyone, my name is Michał Szpak, and I'm an Ensembl Outreach Officer. I'm here today with my colleague Elina, who just joined us as well. And the Ensembl Outreach is a team of about 4 people. We're responsible for delivering Ensembl training worldwide, including longer courses like this course, but also shorter webinars and online tutorials. We're also responsible for the Ensembl help desk, social media, and the Ensembl blog. And today we're going to talk about different data types you can find in Ensembl and how to get the data you're interested in. Let me just switch off my camera for now. All right, a couple of welcome instructions. So this is the course agenda. So I'm going to start today with a short introduction to Ensembl. We're going to talk about genome assemblies and how those generated and produced. And then next time we're going to talk about genes and transcripts. And then my colleague Elina will take over to talk about genetic variation data you can find in Ensembl and the Ensembl's variant effect predictor, which is a really nice tool which allows you to annotate your own variation data. This will be followed by comparative genomic analysis introduced by Emily, and then Elina will come back on the final day to introduce our Biomart, so our online data export tool, which allows you to export large amount of Ensembl's data in bulk. So all of those modules will be consisted of a short presentation where we'll introduce given data type, or a tool will tell you how we produce and generate the data. We'll then have a short demonstration showing you how to navigate the website and how to get the data you're interested in. And then there will be time for exercises at the end of each module, which is a time for you to go explore on your own, try things for yourself. Please remember, it's not a test, it's not an exam, and there are more exercises than there is a time. You don't have to complete all of them. It's just time for yourself to, it's like a starting point for further exploration, and we also encourage you to use your own data and examples if you wanted to. And throughout this course, you will learn what Ensembl is, what type of data you can find in Ensembl, how to navigate the Ensembl browser, and most importantly, where to find help and documentation, as it's just a brief overview. You can find the course materials at training.ensembl.org/event. I'm going to show you that website in a second. You'll find there a copy of this presentation, but also your course book, which contains the demonstration walkthroughs with screenshots and description. You'll find your exercises and the answers. There's also copy sheet, so pretty much just a plain text file with some input for your biomat exercises. So don't worry about that for now. You're going to need that on your final day. So there's basically a bunch of gene IDs and input for your biomat exercises so that you don't have to type anything in. You just copy and paste. And finally, we also have so-called living document, which is basically shared online Google Doc, which everyone can access and edit. I'm going to show you that in a minute as well. And it's a great way of capturing any questions throughout the workshops. You can also use the Zoom chat box. However, Zoom chat box gets pretty messy quite easily and quickly. It's difficult to keep track of. There's no threading. It's difficult to paste any screenshots. Therefore, I would really encourage you to start using the living document. There is a dedicated space for questions, and I'm going to show you that now. So let me just jump out of my presentation and go to my internet browser. You can find the training page at training.ensembl.org. And here you can find information about attending Ensembl event, but also hosting your own Ensembl workshop. And then on the left-hand side, you'll find materials from training section. So let me just click on that link. And here you'll find a list of upcoming Ensembl events. So all you have to do is just navigate to the event you're attending. So here we have Ensembl, a browser workshop, Decode Live. So let me just click here. And on that page, you'll find a copy of this presentation, your course book, and the copy sheet, which I just mentioned. So let me just click on the copy sheet. So as you can see here, have some input for your biomat exercises, which you're going to need on the final day. And finally, there's a link to the living document. So let me just click here. And here we have some welcome instructions and the course goals. If I scroll down, you'll find a course overview and a couple of links, including the link to the teaching materials and feedback survey, which we're going to ask you to fill on the final day. We have some golden rules for efficient online training. So I would ask you to keep all of your microphones muted throughout the lecturing to reduce the background noise, the ambient noise. However, if you'd like to ask a question out loud, it's absolutely fine to unmute yourself. So, in case of emergency, if my screen is not showing or if there is a burning question, I'd rather you to unmute yourself, ask a question right away so we can sort it out before we move on and you're getting more and more confused. You can also ask a question on the Zoom chat box, as I mentioned, and here in this living document. So why the living document is great is because it stays for your future reference. So we won't delete it. So all of the questions and answers captured today throughout this course will stay for the future reference. And you can also easily paste any screenshots. As everyone can edit this document, please watch out and not to accidentally delete other people's content. Furthermore, as we're going to have some practical exercises, I'd like to monitor the progression of the practicals using this raise hand button. So it would be great if we can take a minute so that now everyone can locate this raise hand button. You can find it in your Zoom interface. So depending on the Zoom version you're using, it can be found either under reactions or under participants. So it's going to look like this, a palm of your hand. So it will be great if we can locate this button now and maybe test it. So if you found this raise hand button, please, could you click on it now? I can see 3, 5. Yes, so since a couple of people already know how to use it, that's great. So we've got about 20 hands up. That's really great. So I'm going to ask you to raise your hand during the practicals once you think you're done with your exercises, once you think you've got enough or you addressed all of the exercises you are interested in, so that I can know, I know I can move on with the course. And then we have a couple of resources, so some useful links, and finally, this dedicated space for questions. So please just type your name, type the question, and then me and Elena, all the other trainers will be checking out this document throughout the course, typing in any answers. All right, great stuff. Let me go back to my slides now. So let's move on to introduction to Ensembl. A little bit of history. As you might already know, the first genome was sequenced by Fred Sanger and his team in 1977. And it was a small bacteriophage genome of about 5,000 bases, which paved the way for more complex organisms to be sequenced, including the human genome of about 3 billion bases published in early 2000s. And the advances in sequencing technologies enabled the scientific community to sequence not only many different individuals of a given species, like, for example, various people of different ethnicities to assess the genetic diversity and allele frequency within the population, but also enabled sequencing of many different organisms. And now we have those large international consortia aiming to sequence all living organisms on Earth. And Ensembl was first started in 1999, when the human genome was in commission, as it was known that the automatic gene annotation will be essential. So Ensembl's goal was to automatically annotate the human genome, integrate this data with other publicly available biological data, and making it all publicly available. And since Ensembl launched in 2000, many other genomes have been added to Ensembl as well. So why do we need genome browsers in the first place? Well, essentially, what we're getting from a sequencing experiment is this overwhelming bunch of letters of the genetic code, which is difficult to make sense of and interpret without a context and annotation. So what Ensembl does is that we take publicly available genomes, and I have to stress, we do not do any sequencing ourselves. We only do publicly available genomes, and we make sense of them by mapping genomic features such as genes and transcripts, exons, genetic variants, and regulatory elements to the genome sequences, somehow decoding the genetic information, putting the genome into context. We also perform analysis on that data. So for example, we calculate our own comparative genomic analysis in-house, and we're going to be talking about that later on. And Ensembl is also an added value resource, which means we bring together all sorts of information from various databases inside in a single website. So it's also a great starting point to branch out as well. And we are not the only genome browser out there. We also have our sister page called Ensembl Genomes, which is dedicated to non-vertebrates. And you might have also come across the UCSC genome browser and the NCBI's genome data viewer. So Ensembl is a comprehensive genome browser. Database, that's the word, composed of different types of data. So our primary data type are genome assemblies and gene builds, so set of genes for over 300 vertebrates in the main Ensembl site. But we also have some genetic variation data, not for every single vertebrate in Ensembl, but for a good representation of them, about 25 or so. We also have comparative genomic analysis performed in-house, and that includes whole genome alignments, gene trees, and homologs. And we also have so-called regulatory build, so a set of regulatory elements predicted from experimental data for human and mouse. We also provide some tools for data mining and processing, like, for example, the Biomat, which I briefly mentioned earlier, which is our online data export tool, which Alina is going to introduce at the end of the workshop. It's very handy, allows you to export a large amount of Ensembl data in bulk. We also have tools for data processing, like, for example, the Ensembl ZVEP, which stands for a variant effect predictor, which Alina is going to introduce on a third day, on the third session, which allows you to annotate your own genetic variation data We also allow you to display your own data plotted along the Genome browser, and we provide programmatic access via our APIs. So we have two APIs. We have a Perl API and REST API, which is language agnostic. We're not going to teach those during this course, but we also run a separate course for the REST API access. And finally, most importantly, Ensembl is completely open source and free to use. We have an FTP site, which is a public directory with whole genome flat files for whole genome analysis, and all of our code is publicly available on GitHub. So I already hinted that you can, there are different accession scales. So you can access our data one by one using our browser. And that's what we're going to be focusing on throughout this course. But we also provide a larger scale access. So accessing groups in bulk via tools like Biomart and the web, but also our APIs and directly via MySQL. And we also have this public directory called FTP site for whole genome flat files for genome-wide analysis. So Ensembl is dedicated to vertebrates. We host over 300 vertebrate genomes, and the number of genomes is constantly growing. However, if you look closely, you will realize that we also host a couple of non-vertebrate model organisms in Ensembl, and these include Drosophila. So we've got a fly genome, C. elegans, so the worm, and the yeast species, and they're there for historical reasons, but also for comparative purposes, as they serve as an outgroup to root the tree. We also have important agricultural animal breeds, like, for example, a pig breed and a couple of different mouse strains here in orange. So as I just mentioned earlier, we also have a sister site called Ensembl Genomes, which is dedicated to non-vertebrates, and it's organized by taxa, and all of those different taxonomic divisions are color-coded. So you can find there Ensembl bacteria, fungi, metazoa, plants, and protists. And they all follow exactly the same logic and organization as the main Ensembl website. And here we have a comparison of Ensembl and Ensembl genomes. So as you can see, Ensembl is a little bit older. The main difference here is that we annotate most of the vertebrate genomes in Ensembl ourselves, while for the non-vertebrate organisms, we import annotation from external sources. So the annotation is done in collaboration with scientific communities. So for example, for Drosophila, we would import the annotation from the Flybase. So let's now talk a little bit more about genome assemblies, which are our primary data type, and let's talk about how they're being generated and assembled. So I have to start by saying that there's currently no sequencing technology which allows sequencing of entire chromosomes. Therefore, the genome is often first fragmented, and then the chunks are being sequenced. And I'm showing you here a little bit of a historical approach of how the human reference genome has been sequenced and assembled. So the genome was first cut into chunks, and those fragments were then cloned into bags, so bacterial artificial chromosomes. Those bags were then grown up in bacterial cultures to amplify the number of DNA copies. And then the DNA was sequenced from plasmids using a Sanger sequencing. And obviously nowadays, cloning is not essential. We have PCR amplification, or often there's some sequencing technologies we don't need any amplification at all, so PCR free. And sequencing generates so-called sequencing reads of varying length, depending on the sequencing technology, but in all cases, shorter than the genome. And then the reads are matched up using the overlaps to create a continuous sequence. So how are those chunks, how are those DNA fragments assembled together? So the set of sequenced back clones plotted along the genome is called a tile path. So here those blue blocks represent different clones. You can see they're organized in order, so they're plotted along the genome with some overlaps. And then those back clones with trimmed overlaps are called contigs. And contigs here are shown, are indicated by different shades of blue. So here we have one contig and another, and here we have two more. And then contigs stand together, stacked together with no gaps are called scaffold. So here we have one scaffold, and here we have another. And then finally, scaffolds are assembled into chromosomes. I have to stress that the human reference genome is not a genome of a single person, okay? It's not a consensus either. So every single fragment, every single content of the human genome is a real person's sequence. However, those chunks might come from different people. So about 70% of the human reference comes from a single individual, African American male, referred to as RP11. And then the remaining 30% is derived from multiple people, several people. So in a way, the human reference genome is a hybrid genome of different people's DNA. So it's a composite genome. And people often assume that the genome assembly is a perfect representation of the genome, but I wanted to use this map analogy to show you how it really is. So imagine that the genome, or in other words, all of the DNA within a cell, is here represented as the Earth. Then the genome assembly would be a map, so a representation of it with the coordinate system, some annotation and labeling. And we would like the genome assembly to be as accurate representation of the globe as possible, like for example, Google Maps. However, in the reality, it's something more like this, all right? So it's not perfect. There are errors and gaps. There are genomic regions which are difficult to sequence and reconstruct. There's a lot of repetitive stuff. So with time, the scientific community might fill in the gaps, might correct the errors. Let me see. It's all right, I think someone just joined with the mic on, but that's all right. So scientific community closes those gaps, corrects the errors. So the scientific community might issue some patches, which are those little corrections. And for example, here we patch up Australia and New Zealand. It would be great, guys, if you could keep your microphones muted to kind of reduce this background noise. That would be great. So I hope that will help.
So, Dr. Michael, please unmute yourself. I have muted all.
So the bottom line here is that the genome assemblies do change and are being improved upon, okay? And that brings me to the different versions of the human genome assemblies. And we are currently using the newest genome assembly, GRCH38, which got the shortest gap length, also many rare and private alleles previously found in the human reference have been replaced with the more common alleles. In our GRCH38, data and software are regularly updated. We also host the previous human genome assembly, GRCH37, still widely used in the clinical community. But please bear in mind that there are still some large gaps there. And also, although our GRC37 software is regularly updated, the data there and not so often. And finally, we also host even older Human Genome Assembly NCBI 36 frozen in time in our archives, so it's no longer updated and it's a bit rubbish. There's so many gaps and errors. So this evolution of genomic data, biological data, is also reflected in our release cycle. So Ensemble's data is not static, as genomic data changes rather quickly. So every 3 months, we put up a new Ensembl version. And during that time, there's a lot of behind the scenes work. So we would import new genome assemblies, and we would update our gene set. So we would run our gene annotation pipeline on those new genomes. We would also update our software. We would then rerun our comparative analysis on this new data and update our variation and regulation data. And then finally, we package it all up and display in the browser. So this release cycle and data update implies that Ensemble's data might not be the same if you come back to Ensembl after some time. Therefore, it's important to note which release and number you're working with. If you wanted to come back to exactly the same data to make your analysis consistent, you can find all of our previous releases frozen in time in our archives. We keep our web archives for up to 5 years, and then our FTP archives are kept indefinitely. So the current Ensembl version is 104, and for Ensemble's genomes is 51, both released in May 2021. And on top of our regular release cycle, we also have some so-called Ensembl rapid release, which has a faster turnaround. So what is Ensembl rapid release? It's basically a separate site with new genomes and gene annotations released every 2 weeks. It's got less functionality, and there is no other data types like comparative genomic analysis or variation data. There is no biomart, but you can find there over 200 species now. There's also a BLAST and the FTP site. So it's a great way of quickly accessing new genomes and gene annotations. All right, great stuff. And now we have time for our first demonstration. So before we jump into the demonstration, are there any questions regarding what we've just covered? I can't see any questions in the living document or nor in the Zoom chat box. So I assume there's no questions so far. So let's move on to our first demonstration. We're going to look at the Ensembl homepage and try to find information about the species and genome assemblies. You can find the demonstration walkthrough on page 5. And let me just jump out of my presentation now. And I go to my internet browser. You can find Ensembl at www.ensembl.org, and here we have the Ensembl main page. Would you like me to zoom in, or is that okay? Can you see everything clearly? I assume that's probably clear. I might, just in case, zoom in a little bit if someone's joining us from a smaller screen. So let me just zoom in a little bit. I hope that's a little bit better. So how to navigate this website? First of all, we have so-called navigation bar, which is this blue bar on top, which is always visible. It's always on top on every single Ensembl page. And the Ensembl logo is a shortcut. So it's a link to the homepage. And then we have some other useful links. So we have shortcuts to our popular tools, like, for example, BLAST, the variant effect predictor, the tool section itself, a couple of more tools, and the biomart. We also have the download section where you can find a link to the FTP site. We then have help and documentation. So let me click here. And we have absolutely tons and tons of help and documentation. And here you can find a bit more background information on our data, how it's being processed and produced. And you can find more information about navigating the website. There's plenty of video tutorials. We've got a glossary here and frequently asked questions. So let me go back to the main Ensembl page by clicking on the Ensembl logo. We then have two search boxes. So we have the main search box with a species selector, which is this drop-down list over here. You can choose whether you'd like to search across all Ensembl species or whether you'd like to search a specific genome of interest. Here we have also a section for popular genomes, so-called favorite species. And you can find here human, mouse, and zebrafish. You can also choose to search help and documentation if you wanted to. And you can search for all sorts of things using the main search box, including gene names, genomic coordinates, variant IDs, and phenotype names. We also have corner search, which allows you to search all Ensembl species. And it's a part of this navigation bar, so it's always visible on top in the upper right corner. Then on the right-hand side, you can find the news section. So you can see what is the current Ensembl release and what's new in it. You can find a link to the Ensembl rapid release site we just talked about, and some links to our Ensembl blog. And finally, if I scroll down to the bottom, you can find this tiny link over here in the bottom right corner, which says View in Archive Site. So if I click here, it's going to open a list of available Ensembl archives, frozen in time. So exactly as would they have appeared back in time, ordered chronologically, except the Ensembl GRCA 37 site, which is slightly different from other archives as it's not entirely frozen in time. And there's some occasional data updates. So let's explore one of the archives now. So let me click, for example, on Ensembl 97. If I click here, you'll realize that the color scheme has changed. So you can see now we have this brownish color instead of blue, which indicates you're not looking at the current Ensembl release, but at an archive site. You can also easily navigate through the archives using the URL. So you can see this archive URL starts with jul2019.archive prefix. You can also change this prefix to E97, which stands for Ensembl version 97, and click hit. And that should take me exactly to the same site. So let me go back by clicking the back button in my browser. We then have the genomes section. So you can navigate to the species homepages from here. And we also have a section for favorite genomes, which includes our popular genomes, such as human, mouse, and zebrafish. There's also a link to the GRC's 37 page here. And you can add it to your favorite genomes if you wanted to do so. You'll have to log in to your own Ensembl account here at the top of the page. So this section also contains the species selector. So you have a drop-down list of all available genomes, and you can navigate to species homepages from here. Alternatively, you can explore the full list of Ensembl species as a table. So I'm going to click on this view full list of all species now, which takes me to this all species page, where you can find a table listing all of available genomes. You can find the common name, the Latin name, the taxon ID, but also the genome assembly name and accession number. You can find the gene annotation method and availability of variation and regulation data. So for example, a Y here stands for yes, that means we have some regulation data for this given genome. You can further manipulate this table. You can sort it using those little arrows in the table header. You can download the entire table by clicking on this Excel sheet icon, and you can filter it using this search box. So let me search for my species of interest, for example, human. And then I can go to the species homepage by clicking on the species common name. And that takes me to the species homepage. Here you can find a species specific search. So if I search for anything here, my search results will be restricted only to human. We then have some example pages introducing a different available data types. And most importantly, we have this genome assembly section where you can find more information about this genome assembly. So you can see we're currently using GRCH38. You can find the patch version after the decimal point. So we're currently on patch 13. And here we also have the genome assembly accession number. There's also a list of previous genome assemblies for this given species. So if I click on this drop-down list under other assemblies, that gives me a list of older assemblies still available. That includes GRCH37 and NCBI 36. And I wanted to bring your attention to this more information and statistics link. So under this link, you can find more information about this specific genome assembly and the gene annotation. So let's explore it now. So let me click on this more information and statistics link. And here we have a bit more info about this genome assembly and the gene build. It tells you who generated it and how. You can find the gene length. So we're giving you a so-called golden path length, which is basically a one linear representation of the genome, which only includes the primary assembly. It doesn't count alternative sequences, so assembly exceptions such as haplotypes and patches. So we already know that patches are those little updates or corrections, and haplotypes are basically different versions of the same sequence. You can find the current gene annotation version, which is gen code 38. We're going to talk more about the gene annotation in our second module. And you can find some summary statistics, so gene counts for both coding genes, pseudogenes, and non-coding genes on both primary assembly and alternative sequences. Great stuff. So I wanted to quickly show you the Ensembl Genomes homepage as well. So if I type Ensembl genomes.org and hit enter, that should take me to the Ensembl Genomes homepage, which is composed of those taxa, division pages, which are color coded. So we have here the Ensembl plants, metazoa, protists, fungi, and bacteria. You can also find our COVID-19 portal, which hosts SARS-CoV-2 genome and gene annotation. And there's also a link to the Ensembl rapid release. And I wanted to quickly show you the Ensembl bacteria page, as is organized slightly differently due to high number of bacteria species that we host. So let me click on this, go to Ensembl bacteria link. Let me maybe just zoom in a bit. So it follows exactly the same logic and organization. There's only one change, which I'm going to show you in a minute. But first, I wanted to bring your attention to this little arrow next to the Ensembl bacteria logo. So you can easily navigate through Ensembl bacteria using this drop-down list. So you can go back to the Ensembl Genome's main page, but also other divisions. And you can also go back to the main Ensembl page dedicated to vertebrates if you wanted to. You can also easily navigate through Ensembl genomes using the URL prefix as well. So the difference which I wanted to point is that you can also find a full list of Ensembl bacteria species here, but it's really, really vast as we host over 30,000 of bacterial genomes. So instead, we also have, you can find two searches, one to search for a gene across all species, and another one to search for a genome of interest. So let me, for example, type Clostridium. And as I type, I should get this autocomplete suggestion with this drop-down list. So let me click on any of those to go to the species homepage. And here we are. So it's exactly the same page that we saw earlier for human. Great stuff. So now we have time for a set of exercises exploring what I just showed you. So genome assemblies and species homepages. Are there any questions before I move on to exercise? I can see there's a couple of questions. Do you have genomes for plants too? Yes, so you can find the plant genomes in Ensembl Plants, which I just showed you. What the regulation data means. So we're not going to be getting into details when it comes to regulation data throughout this course, but regulation data is basically prediction of regulatory elements, things such as gene promoters, enhancers, and transcriptional repression, and so on and so forth, transcription binding motifs, which are predicted from experimental data. So for example, from ChIP-seq experiments and histone modifications and so on and so forth. So we do that for human and mouse. I'm sorry, someone just wanted me to repeat something. I'm not entirely sure what you're referring to, but if you can type a specific question in the living document, I could address it. When it comes to the video recording, I believe they will be made available by our host, so please contact them directly. So Ensembl, someone asked, do Ensembl provide primal design tool like NCBI? Unfortunately not, we don't provide any primer design tools. We provide genome sequences or gene sequences, which you can then use in your design, but we don't provide tool for a primer design specifically. And someone asks, what are the differences between GRC 37 and 38? So as I briefly mentioned earlier, GRC 37 is a little bit older, so there is a longer gap length. So some of the gaps were filled in GRC 38. So that's like the biggest improvement. And because of that, you might find differences in genomic coordinates for specific genomic regions. There might be more genes annotated on GRC 38 as it's more complete genome assembly. So it's basically newer and more up to date. Does Ensembl help in next generation sequencing data analysis? In some ways, yes. So once you've got your variant calls, you can then analyze them using the variant effect predictor. So that's sort of like the last stage of next generation sequencing analysis. So once you called your genetic variant, you can then run it through VEP to sort of add a meaning to them. So annotate them and see how they could potentially affect genes. All right, so as I mentioned, now we have time for the exercises. So you can find your exercises on page 10 in your course books. So if you navigate to the training site, which I just showed you, let me maybe just copy paste this link in the Zoom chat box. So if I copy this link and let me just paste it here. If you navigate to your course books, you'll find the table of content, and here we have the section exercises, ensemble species. Have a look, give it a go. Again, please don't feel like you have to tackle all of the exercises. Please choose based on your interest, whatever feels relevant to you and your work, whatever fits your research questions and use cases. And let's have about 10-15 minutes for those. Choose one or two, give it a go, and then once you're ready, please raise your hand so that I know we can move on to our second demonstration. And please feel free to keep asking questions. It would be great if you could put them in the living document. So while you're working on the exercises, I'm going to be typing answers there. And if anything's unclear, if you need any assistance and help with the exercises, please do let me know. I'd be very happy to explain the exercises on the screen. We can go over the answers all together. All right, so please feel free to start now, and I'll see where we are in about 10-15 minutes. So someone asked how to go for exercise. So if you go to this link, I just pasted, and then you find the PDF with exercises. Yet again, it's just time for you for your own exploration. So you can find the exercise, a text over here. So you've got a couple of exercises you can choose from, and then try to solve the puzzle. So we give you some sort of question that we would like you to find answer on your own, that will encourage you to explore the ensemble pages. Yet again, it's not a test, so we're not checking your answers. It's not an exam. And if you cannot, if you don't know how to get to an answer, please let me know, and I'll be happy to give you the answer or walk you through the answer. You can also find all of the answers in a text form after the exercises. So someone asks whether it's possible for analysis of non-coding RNA in Ensembl a genome browser. So we provide non-coding RNA data. We also perform some analysis on that data, like for example, non-coding RNA gene trees. However, yet again, genome browsers are more about browsing the existing data produced by us rather than performing your own analysis. You can also obviously use the files, the references files that we provide and certain tools to mine the data and so on. But if you ask me, if you tell me a little bit more about the analysis you're thinking of, if you're more specific, and that will be helpful. So someone asks about Panda. I'm not entirely sure what you mean though. So someone, yeah, so after completing the exercises, no, you don't have to send them anywhere. That's absolutely fine. So if your answer matches the answer in the course book, that's great. You can move on. I'm going to go over the zebrafish exercise in a second. Okay, great. It's great that you help each other. So I'm really glad that you are very active on the Zoom chat box and the living document. So let's have a look at some of those exercises. So I feel like someone has already answered it in the chat box, but basically, so we are asked to go to Ensembl Metazoa. You can go there directly. You can just type Ensembl Metazoa in Google, and that should list a link to our Ensembl Metazoa. You can also go there from Ensembl Genome. So if I type Ensembl genomes.org, you'll find a list of all of those taxa divisions. So if I go to Ensembl Metazoa, that will take me to the homepage, and then we're asked to explore a different mosquito species and genome assemblies. So if I go to this view full list of all species, that's going to give me this table of all available genome assemblies, and we're interested to count number of assemblies of this specific genus. So let me just paste it in the search box, and that should filter my table. And now you can just simply count how many of them we've got. So I hope that explained it. Let me just have a look at the zebrafish exercise for you as well. Yeah, so the exercise asks what previous assemblies are available for a zebrafish. So if I go to too many windows. If I go to the main Ensembl page from here, so I can click on this tiny little arrow next to the Ensembl Metazoa, and I can go back to Ensembl Vertebrates. And then you can find a list of available genome assemblies in the species homepage. So if I go to zebrafish homepage by clicking on the zebrafish name, you will find the list of other assemblies available here in this drop-down list. So you can see, apart from the current genome assembly, there are also 3 archived assemblies available here. Someone asked about the panda exercise. That's right. The full species name is a giant panda. All right. I hope all of the answers here on the chat box were answered. How are we doing with the exercises, everyone? So I can see 12 hands up. Do people need more time? Oh yeah, I can see now a few more people are done. We'll be moving on shortly. All right, as we don't have that much time left, I think it would be best to move on to our second demonstration, and then you'll have some more time for the exercises later on. So let's go back to my slides now. So we're going to have the second demonstration now. So we're going to look at a region of human genome. And we're going to manipulate to view to see the data we're interested in. You can find the demonstration walkthrough on page 11. Let me just jump out of my presentation. I'm going to copy this genomic coordinates, and I'm going to go to the Ensembl browser. So let me just go to the homepage by clicking on the Ensembl logo. So I could search for genomic coordinates using the main search box. So I can just type my genomic coordinates over here. And as genomic coordinates are ambiguous, so you can find exactly the same genomic coordinates, for example, in human and mouse. So that's not specific enough. We need to tell Ensembl which species we're interested in. So I can type human before the genomic coordinates. Alternatively, I can choose the species from the drop-down list to restrict my search results only to human. I wanted to bring your attention to the genomic coordinates format. So we first have the chromosome number, and you don't need the CHR prefix. You could use it. Ensembl is going to understand it, but you don't need it. So just the chromosome number. We then have a colon, start position, hyphen, end position. As you can see, there is no white spaces, there's no commas. Yet again, Ensembl would understand it, but you don't need that. Just the chromosome number, colon, start position, hyphen, end position. And once ready, simply click go. That will take you to the location tab, region in detail view. So Ensembl data is organized in a tab system with the different tabs dedicated for different data types. So the location tab is the first tab we're exploring throughout this course. And it basically allows you to browse the genome and scroll along the chromosome. So I first wanted to point out the the page specific help. So whenever you're in troubles, you get lost, you don't know how to do something, look out for this question mark symbol. So question mark symbol indicates this page specific help. So if I click on the question mark, it's going to open a new window with a bit of explanation. You have screenshots with labels. You also have frequently asked questions, video tutorials, a glossary, and contact help desk link. So please feel free to drop us an email whenever you're in troubles, you're struggling with anything, you don't know how to get around, drop us an email and we normally reply within 1 or 2 working days. So let's close this windows for now. We then have the page specific menu in the left, which lists all available views on this page. It helps you to navigate through this page. You also have some links to other external genome browsers. We then have so-called tool buttons below. So these are the blue buttons over here. The first is configure this page, which allows you to customize this view, to add more data to this view, as by default, we only show you a small amount of data. There's way more to explore, and we're going to do that in a minute. We then have the custom tracks blue button, which allows you to upload your own data to be visualized, so to be plotted along the genome. We then have export data button. So if I click here, it will open a new window, which allows you to export data for this particular genomic region. You can output either either sequences FASTA, or you can also export genes for this genomic region using one of the feature file formats. So you can export genes as comma separated files, tab separated files, but also a GTF, GFF, and BET files. You can further manipulate and change these genomic coordinates and the length of the flanking sequence. To exit this window, you simply have to click on the tick or anywhere outside the box. So let me just click on the tick for now to close this window. And then our main view over here is composed of 3 images of different resolution. next more detailed than the last. And we're going to go over them one by one now. So our first image is so-called chromosome view, which shows you our current location on the chromosome. So our region of interest is indicated by this red box. So you can see the chromosome diagram with cytogenetic banding. So the cytogenetic bands are shown as those blocks of different colors, white, gray, and black. And then the alternative sequences, so assembly exceptions such as haplotypes and patches, are shown as those red and green boxes. So if I click on one of those, it's going to open a pop-up menu with a little bit more information about this specific patch. So you can find the patch name, you can find the genomic coordinates, you can compare this alternative sequence with the reference, and you can learn more about the assembly exceptions. So patches come in two types. Green are fixed and red are non-fixed. Furthermore, I can also draw boxes in this view, and that will give me an option to either mark this region, so highlight it, or jump to the new selection. Our second image is so-called 1 megabase region. It's also centered around our region of interest. So you can see our region of interest is still the red box, and we can see a wider genomic context. So we can see 1 megabase around our region of interest. Data here is organized in tracks. So what is track? Track is this very genome browser specific term, which is basically a type of data plotted along the genome. And you have different tracks for different data types. Here we have a track from chromosome binding. Here we have our genome assembly track named contigs. So this is basically our double-stranded DNA, and contigs are here shown as blue boxes of different shade. And then we have another track for genes, which are here shown as blocks. And you can find the gene name under the block, and the gene orientation is indicated by this little arrow next to the gene name. And we also have a track for a regulatory build, so a regulatory elements. And you can find the explanation of those colors in the legend below. Furthermore, you can scroll along the chromosome in this view using either those scroll arrows. You can also draw boxes in this view and jump to a new selection. Furthermore, you can change the mouse action. So here you can see you have 2 mouse actions available. Select a region, which is currently active, but I can change it to scroll, which allows me to scroll by clicking and dragging. And as soon as I change the genomic coordinates, the image below grays out and gives me 2 options to either update this image to new coordinates or a reset configuration. So I don't want to change my genomic coordinates now, so I'm going to reset. And then our final and the most detailed image is basically our red box. So that's our region of interest. And data here is also organized in tracks. You can see the genome assembly track, also named contigs, and genes are shown as transcript rather than boxes. So a gene is a set of transcripts in Ensembl, and the gene track is split into 2. So you can see forward-stranded genes above the genome assembly, and reverse-stranded genes below the genome assembly. You can also see that those tracks are shaded differently. So reverse-stranded tracks have this peach highlight over here, while the forward-stranded tracks have this dark blue highlight. And then tracks which are not strand-specific are shown in this light blue color. So for example, genetic variants affect both DNA strands, so they're not strand specific, and therefore they're unstranded, so they're shown in this light blue color. Furthermore, all of those features are interactive, so you can click on them, and that's going to open a pop-up window with more information, hyperlinks to other pages. You can also draw boxes in this view. And for example, let's say I'm interested in this exon. So exons are shown as boxes connected by introns shown as lines. So if I wanted to highlight this exon, I can choose this option to mark a region, and that's going to create the highlight, which is going to stay visible even if I zoom in or zoom out or I change my genomic region. If you wanted to get rid of the highlight, you can simply click on this tiny menu over here. So if I click on it, that's going to give me an option to remove the marking. Alternatively, I could also click on this option over here, which says clear marked region, to get rid of the highlights, which I'm going to do now. Great stuff. So let's add more data to this view. If I scroll up and go to configure this page, that's going to open a new pop-up window with which lists currently active tracks. So here we have all of the tracks which are currently active. If you don't know what any of that means, you can simply click on this information icon to have a bit more information about those tracks. You can manipulate this view, you can change the decoration and styling. So you can also turn tracks on and off. So for example, if I click on this box, that's going to turn the sequence track off, but I don't want to do that. I'm going to bring it back on. Some tracks come in different styles. So if I click on this genes track over here, it's going to give me a pop-up menu with all available styles. And finally, as we by default show you a small amount of data, there's way more to explore. You can find all of the data available grouped into categories in the menu on the left. You can also navigate through that list of tracks using this search box, which can help you to find the tracks which are relevant to you and your work. So let's say I'm interested in UniProt proteins. So I'm going to type UniProt, and I'm going to turn on the proteins mammal from UniProt. So I'm going to click on this box and turn them on as labels. Furthermore, let's say I'm interested in exact variants. So I'm going to type exact and turn on the exact variants as normal. Once you're ready, choosing your options, simply click on the tick, save, and close. And that's going to update my view. Let's just give it a second. Sometimes it might take some time depending on the internet connection and the traffic on our website, but hopefully it's not going to take too long. I'll have a look at the questions meanwhile. All right, so here we have our newly added tracks. I'll get back to questions in a second. So you can see here we have our proteins from UniProt added as a new track. And proteins are also split into two tracks. So we've got forward stranded proteins above the genome assembly and the reverse stranded proteins below. We also have the newly added exact variants. Those tracks behave as any other track. So you can click on the track name to also further customize this track in this view. So you have this cogwheel, which allows you to change the track styling. You can also get rid of the track if you're done working with it. So let's say I'm going to get rid of this. Furthermore, you can also click on the track and drag it around up and down. So you can arrange this view as you want it. And then once you're done arranging it the way you want, you can share it with your colleague or collaborator. So if you click on this, share this page link, that is going to generate a custom link, which remembers all of the information you added and the way you arranged it. It also remembers the release number. So if you send it to your colleagues and they open it in a couple of months, it's going to take them to our archives. Finally, you can also export this view as an image. And if you wanted to go back to default configuration, you can do it by clicking on the reset configuration button. And the last thing I wanted to show you can alternatively reset configuration by going to configure this page, and you'll find their same button which says reset configuration. Okay, great stuff. So, and now we have the last couple of minutes for your own exploration. You can find the exercises on page 17 in your course books. And let me now have a look at the remaining questions. So someone asks about cytogenetic banding. So this is, I'm sure you can look it up. It's basically concepts, something to do with a chromosome staining. So, it's this phenomenon which has been discovered, years ago, really, that different a chromatin of different density will stain differently. So if you stain a chromosome with certain dyes, you'll see this a banding pattern. And someone asked about patches in a chromosome view. So let me just jump back to the browser. So if you look at this chromosome diagram, you'll see those little boxes over here. And they represent those alternative sequences, so assembly exceptions. So on top of the primary assembly, you might find patches, which are those little updates and corrections, and haplotypes, which are basically alternative version of a given sequence, as some genomic sequences are difficult. Variation in certain genomic sequences is difficult to represent using one linear representation of the genome. That's why sometimes we have those alternative sequences. And you can find more, you can learn more about them by clicking on the box and then choosing what are assembly exceptions. And then patches can be either fixed or not fixed, and green boxes represent the fixed patches. So we still have last 3 minutes, literally. Now we have this time for your own exploration. And then in about a minute or so, I'm going to wrap up for today as we're slowly running out of time. So we're coming to the end of this session, but I'll be back in a minute or so for a final closing remarks. And I'll tell you what to expect from the next session. All right, everyone, as we literally got a last minutes left. If there are any outstanding questions, if you would like to carry on the exercises in your own time, you're more than welcome. Please feel free to keep adding any questions to the living document. I'll be responding to all of those questions there. And we can also address any outstanding questions regarding those exercises during our next session. So if there's anything unclear, I'd be very happy to go over any of the answers on the screen at the beginning of next session. And I'll see you all on 22nd of June. We'll be talking about genes and transcripts in Ensembl. I'll tell you how we annotate our genes. And thank you very much for joining today. Thank you for thank you to our host for having us today and facilitating this very nice session. And I'll see everyone next time.
So, so thanks, Dr. Michael, for such a wonderful and energetic session. We are honored that you delivered on our platform. We will meet again on 22 again with Dr. Michael. Topic will be genes and transcripts. Hope you people enjoyed learning, and I will share video recording of the session as soon as possible. So with these words, now I seek permission of Dr. Michael to end the session.
Thank you, everyone. Goodbye.
Okay.